And now I'm going to share screen. Share. And go to Chrome. Okay, here we go. So let's think about molecular pathology. So I think these are probably some things that you should be able to at least be able to define or use in a sentence. PCR, DNA sequencing, DNA polymorphism analysis, we'll forget about those, gene linkage analysis, yeah, SNPs, genetic association studies, and FISH. I think those are the important ones. So let's think about some principles here of sending things for pathology testing. And it particularly also applies to molecular testing. So the specimen type, the limitations of the test, and the clinical value. Now, this might seem very easy, but it is very important. So, specimen type, right. So what specimen do you send? So the first is that if you are attempting a carrier type, these cells have to grow, and so you have to send it fresh. You can send it using blood or cheek swabs for germline mutations, carrier typing, or more in-depth genome stuff, genome sequencing, but it needs to be sent fresh. Okay. For infectious cases, you should also send it fresh. Okay, so fresh tissue for carrier typing, for looking for germline mutations, and looking for infectious disease. And usually, when we're looking for mutations, we send blood or cheek swabs. We can use formalin-fixed tissue for most DNA studies, such as FISH and mutation analysis, but it's not as sensitive as the fresh tissue. So remember we talked about sensitivity and specificity. So sometimes we can attempt to do gene things, but unless the test is really robust, we might not find it. So if in doubt, you're going to do something. You're going to go and look up what specimen to send. Okay, your laboratory, so I work with Capital Coast DHB, will have a test specification document, and it will tell you exactly what to send. So it's very important that you send the right thing. We're all experts now at microbiological testing because of COVID. So as you know, if you do a throat swab rather than a nasopharyngeal swab, you won't pick up the COVID as well. Different specimens need different stuff happening to them in the lab. So this is a test that could be valid for blood, won't be valid for a nasal swab, for example. You won't be able to get DNA from a fixed sample, but you will be able to get it from a fresh sample. So results from a non-validated specimen will not be reliable. So please send the right, this is principle number one, send the right specimen to the lab, and you can do that by looking it up on the lab record. Mostly, if you're in doubt, send it fresh. Number two, clinical details. So we have a policy at CCDHB, no details, no lab test. So you have to know why you're ordering the test, okay? That will help you think about what specimen to send. So know what you want to send. Limitations of the test. So all tests have limitations. You need to make sure what it is actually detecting. So we talked about before, will it detect all variants or the right variants? So telling a pathologist that they must do a BRAF melanoma mutation on a uveal melanoma is not going to be useful. The BRAF melanoma mutations are used doing PCR on a machine called a cobas machine, and it only detects the mutations it's programmed for. So even if it comes back saying no mutation detected, it will say no mutation is detected and these specific mutations have been tested for. So all genetic tests have limitations. Can tests detect antibiotic sensitivities? No, it will detect the bug, but can't do sensitivities. So if you want to do sensitivities, then you've got to send a regular specimen so we can grow it. Is it an active infection? We all know that DNA knocks around the body for a long time because we've been watching the careful old cases report come through our COVID screening. So they can be overly sensitive and detect DNA when the infection is not still live. So those are the limitations of many tests. Okay, so we have the right specimen type. We understand the limitations of the test. And now we want to determine, is the test of clinical value? Okay. Oh, I'm sorry, we're still on limitations. Another limitation of the test is the limit of detection, the LOD, the lowest actual concentration of DNA that can be consistently detected. So for COVID testing, for example, there will be people who've been cured and have a bit of DNA and they don't even get a false positive because their DNA level is too low. Now, if we're looking for another infection, maybe that DNA level is too low, we won't detect it. But if we're looking for a mutation in a tumor, for example, and we just have a little tiny bit of tumor on a slide, on a big slide, we won't be able, if we put the whole thing through, to detect the abnormal DNA. So usually we need a minimum amount of DNA that has to be present in a sample for it to be consistently detective. And the limit of detection describes the concentration that falls at. Now, with COVID, they've been doing what's called real-time PCR. All right, so in PCR, you know that the DNA is put in the black box and it keeps replicating up until there's lots and lots of copies. And so if it goes through one cycle, it doubles it, and 2 cycles, it 4 times it. And so the lowest concentration that it's detected at will give a semi-quantitative answer as to how much DNA was there in the first place. So we stick the COVID swab in the machine, and if it detects the DNA after 20 cycles, we know that patient was loaded with COVID DNA. But if it only detects it after 30 cycles, we know they hardly had any, and it was an old infection. So the limit of detection determines the analytical sensitivity. Okay, so limitations of a test. There could have been insufficient DNA. Maybe there was a false positive because it was old DNA rather than a live infection. Maybe the controls went wrong, or maybe there was cross-contamination of the sample at the point of collection. So there's lots of things that can influence how accurate a test is. All right, so the difference between analytical and diagnostic accuracy. Okay, what are the distinctions? In a clinical setting, we want to not miss cases, right? So we optimize for sensitivity. If you've got someone with a suspected aspergillus in the eyeball, you want to detect 100% of these cases very, very rapidly, okay? So we want to give it that antibiotic to prevent any further damage happening, okay? In a public health setting, we want specificity because we don't want to isolate all these people. And of course, sensitivity and specificity can have a trade-off. Okay, so we want a high sensitivity test, okay? Yeah, so I'm sure you know the difference between sensitivity and specificity. A high sensitivity test is identifying those patients with the disease, and a high specificity is able to detect patients a true negative. Okay. How will the results help? So let's think of our principles. We want to know why we're doing it. Will it improve patient care? Is this the right test or combination of tests? Can I interpret it accurately if it comes back? Will it influence management, and are there harms? And this is particularly the case for genetic testing. We have to very much consider the harms of testing. Molecular testing for infectious diseases, not so much. Okay, so these are some of the things that we need to think about always. What specimen? The limitations of the test, particularly the analytical sensitivity, the LOD, limit of detection, the diagnostic sensitivity and specificity, and the actual clinical value of the test. I'm going to stop sharing for a moment there. All right, who's awake? Are we all awake? I'm going on some practical examples now. And I see Amanda's been answering in the text there. Excellent, Amanda. Is there anyone else who wants to raise specific questions about principles of testing?
I think the first thing that you brought up about consulting the lab test guide is a very good one. We also have one at Auckland DHB. You don't even have to be in the intranet to look at it. It's just called Lab Plus Test Guide. You can Google it and then you can see the specifications of the test.
Okay, I'm going to do cases now. Now, there are general cases. These are not eye cases, but they will show some principles. I will now share my screen again. And share, and back to Google. Right, here we go. So we're going to look at two cases here, lung cancer and some pharmacogenetics. Right, and at the end of this, we want to be able to describe PCR and FISH, be able to correctly submit specimens for PCR and FISH, understand the reports and the implications of the results. So here we go. We've got a 30-year-old man who had a lung mass and he had some enlarged lymph nodes. We did a biopsy and we decided that this was metastatic adenocarcinoma in his lymph nodes. We did immunohistochemistry. Remember, that detects proteins, and the immunohistochemistry helps us decide where it's from. And in this case, it was from the lung. So we know that lung cancers have a drug that is effective. So non-squamous cell carcinomas of the lung have a possible treatment. So we have a clear clinical indication for doing a test to find out whether a drug will be effective. So the first thing we have to do is to detect whether or not any mutations were present to see whether or not that drug will work. EGFR mutations mean that the patients respond to erlotinib. So the technique we do that is real-time PCR. PCR is good for looking for specific mutations that we know already exist in a population or a tumor, because, okay, it puts in little probes. We're just going to skip over that, okay? So it uses, again, this cobas machine to look for specific mutations. Okay, so PCR can look for multiple but specific mutations. Okay, real-time PCR is the same as PCR, but we can detect the number of cell cycles that it takes to find the result and get a semi-quantitative analysis. So PCR mimics normal DNA replication, right? You put in DNA, you denature it, you put in a starter, right, and anneal it together. Then you put in enzymes and extend it, and then you've got 2 bits of DNA replicated between the start and the finish of the sequence that you're testing for. And then you repeat, and you get lots and lots of that type of DNA. Just that little sequence is amplified. Okay. So we can do that on formalin fixed paraffin embedded tumor, which contains at least 10% tumor. So that is the limitation of detection. Yeah. I'm not going to show you this, but okay. I'm going to show you the lab report. So we have EGFR mutation not detected on this patient. So that means that presuming 10% of the tumor was submitted, within that limited range of mutations that are the commonest mutations, it wasn't detected. Okay. So then what we do with lung cancer is we then do another reflex test for a thing called ALK. Okay. So the ALK rearrangement is a target for a different drug. Okay. So we know that patients with the second mutation will respond to a different drug. So ALK is anaplastic lymphoma kinase and is also present in adenocarcinomas. And this time we're going to use FISH. So why are we going to use FISH this time? So we're looking for something slightly different. Okay, in tumors that have an ALK abnormality, there isn't a single point mutation change that can be detected by PCR, a little probe in a DNA sequence detecting point mutations. We're detecting wholesale abnormalities of the genes. So in the ALK gene, what happens is there is a break in the DNA and we get a chromosomal translocation. So FISH can detect big things, big rearrangements. And in this case, what we do is we label a piece of chromosome either side of where the break point occurs with a red and green probe, okay? And if a translocation has occurred, then we will see separation of those probes. Now, the paired signals often have a kind of yellowish appearance. And so if there's been a break in the FISH, then we'll get one yellow signal and one red and one green split signals. So FISH break apart probes will detect a rearrangement of the chromosomes. Okay, so FISH will only show, so FISH is where we do attach a DNA sequence to the chromosome. Yeah, it binds only those parts of the chromosomes that have that sequence. So it's quite specific to finding a little spot on a chromosome. We look at it down a fluorescence microscope and detects things at the level of whole chromosome changes. Okay, so here this was the FISH and an ALK rearrangement was detected. So the signal, the red and green signals split in some cells, and therefore we were able to detect the ALK. Okay, so limitation of the test, sufficient sample was sent and no other limitations were observed. And there they get crizotinib rather than erlotinib, and we can do that. Okay, so I'm going to stop sharing again. Are you clear about EGFR? Sorry, are you clear about PCR and FISH and what they're used for in this specific tumor?
Can I just ask, with the FISH, so you're still, your probe is still binding to a specific DNA sequence.
Yeah.
How, and you're saying it's sort of like a big picture change, like a big translocation or something like that. So say you had one base pair that was different in a whole string of DNA, would the FISH still bind to that in the same way that it would bind to a normal DNA sequence with that normal base pair?
Yeah.
Is it a kind of bigger picture than that?
Complementarity is too high if there's just one base change.
Sure.
So if I was wanting to see if there was monosomy 3 in a ocular uveal melanoma, what technique would I use?
FISH.
I would use FISH, wouldn't I? A whole chromosome thing. If I wanted to see whether there was a specific GNAC mutation in that sample, what technique would I use?
I presume PCR.
I would use PCR because it's looking for a specific mutation.
Mm-hmm.
Okay. Hopefully that's made it clearer for those two. Are there any questions about PCR and FISH? How does monosomy 3 look like down a microscope in uveal FISH testing? So imagine you've got a probe that attaches to the centromere of chromosome 3, right? And it fluoresces bright green. You pop it on the cell, and there are going to be cells with 2, yeah, or cells with 1. So you look and see whether the cells have got 1 green dot in them, or whether they've got 2 green dots in them. And that will tell you whether there's monosomy 3. Can you do a karyotype to look for monosomy 3? Well, theoretically, yes. However, you would have to submit fresh tissue to the lab to do that, because karyotypes are about growing cells. And so we'd have to know it was a melanoma, and we'd have to take a little bit out and karyotype it and grow it. Now, technically, if you were like about to pop a plaque on a melanoma, you could get a little bit out and send it off fresh to be grown, or we could FISH it. Now, karyotypes take a couple of weeks. FISH can be done in 24 hours. It's faster, but if you want something karyotyped, it's got to grow. Any other questions? Okay, I will just go. I'm not going to do the second half of the lecture because I don't think it's applicable. I am instead going to just go to the second molecular lecture.
One of the other problems with karyotype is if we're collecting fresh tissue, or you don't know if it's actually necrotic tumor or live tumor. If you see a necrotic tumor, it's not going to grow. And a lot of labs have trouble, cytogenetics labs, growing tumor. They're much better at growing fetal cells.
Okay, so let's have a look here.
Karyotypes are good if you don't know what the mutation is, because you're looking at across the whole chromosome pattern. You don't know what it is.
But if you know what it is, it's best to do a targeted test to look for presence or absence of that particular thing, like monosomy 3.
Okay, I'm just having a look at the second lecture. I think I've covered most of the principles actually in there. So yeah, so genome-wide testing is used if you sequence if you don't know the mutation. So for example, if we had a retinoblastoma and we wanted to know the specific mutation in that retinoblastoma, because each mutation is very individual to each of the families, then we would take a bit of that retinoblastoma and we would sequence that gene. And then we would know the specific mutation and could go and look for it in the blood of the person and family members. If we had someone who had was having eye problems because they had a mitochondrial myopathy, we would send cells down to a lab and they would sequence the specific genes that are associated with mitochondrial myopathies, because each one is quite different. So sequencing is used for discovering new mutations. PCR is used for finding mutations when we know what we're looking for. And FISH is used for whole chromosome shifting around abnormalities. Do you need fresh tissue for GWAS? Amanda?
What's GWAS?
Genome-wide
Association.
GWAS. Hang on, let me just find out what GWAS stands for.
Genome-wide.
Genome-wide association, no, genome-wide. GWAS is what they do, genome-wide association study, which is used to associate specific genetic variants with specific diseases. And it involves scanning the genome from many different people and looking for genetic markers. So usually it's done on blood.
That would be like exome sequencing, yeah?
Yeah, so whole exome sequencing and GWAS sequencing, they would need to have fresh tissue. I suspect it can be done on formalin-fixed tissue, but it would have a lower sensitivity, because I know that when I think they do GWAS in our lab, but they've got to grow up lots of like copies before they can use it. So they like to have it fresh. But I suspect it can be done on good solid bits of tumor as well. So I would say either for GWAS. So Mark says, yes, you do for GWAS. Is microarray used in routine practice? So it is used in routine practice, yes, to detect not the massive type abnormalities, but the smaller type structural abnormalities that we find in chromosomes. It's used for microarrays. So I know microarray is used particularly, I think, though Amanda's going to be the expert on this one, for children who have abnormal development and you're trying to find smaller structural abnormalities in chromosomes.
You know, funny looking fetuses, funny looking kids and developmental delay.
Yeah, so microarray is used in routine clinical practice, but not usually for eye specimens.
All right.
We're nearly at 5 o'clock here.
Yes.
I have popped some Anki cards there if people want to do the Anki cards. I know some people like Anki and people don't like Anki. Never used Anki cards before. Now's probably not the time to immediately start. You probably have your other methods of learning.
There's supposed to be answers on your Anki cards, because I had a look and there's no answers. I think you just have to look them up yourself.
That was because this was a project, because you're meant to fill them in as you went along.
Yes.
Oh, okay, that makes sense.
It's called active learning.
So I was providing the prompt question and you guys were providing the answers. Yeah, to help you initially learn and then revise.
Okie dokie. Cool.
Okay.
Time for survey before farewell.
Let's do the survey before we say goodbye to you all. So we really appreciate you doing our surveys, and I will pause the recording while you do the survey, Amanda.
So that's retrieval practice. Spaced repetition. So that's what Anki cards or flashcards are good for. So you do the endo-ophthalmitis on Monday, and then you're going to do revision of your gaps of that on Thursday. So it's the repetition of the bits that you missed out.
So you can use those things that I showed you on line for that, those cases. If you've learned cornea on Monday, then see if you've remembered it on Thursday using
Yes.
the kind of the lectures.
And what we're doing here is, like, if you use the ophthalmology case review book, you're looking at the picture and then you are retrieving it in words. That's called dual coding. So dual coding is an effective way because you're making multimedia going into your brain. So it's both eyes and text written. You can even speak to yourself about it. So that's why it's good to make a diagram of complex interactions like the immunology. Get your whiteboard marker and draw on the bathroom mirror so you see a diagram as well as write the words. So that's called dual coding. We've done retrieval practice. We've done elaboration. Elaboration is description of what you've seen. That's why you have the 6-part answer. You are elaborating. So that's elaboration. What are some other ones that we've got? There are 6 learning strategies. And concrete examples, what we've done to all throughout our course, particularly with the slides, is concrete examples. They are real examples set in a case vignette. It's just what you really do, and it's just what we really do, concrete examples.
So you'll remember it much more with these concrete examples, but if you were only going to do one thing, it would be active recall. Never ever just read the textbook, always test yourself by answering questions, by looking at the slides, and do that ideally a few days after you've learned it.
It's called test enhanced learning. Another trick to do is to do it before you go to bed, because the modifiers of learning are sleep and exercise. So you do the hard thing before you go to bed, all the things go around into your mind. They go from the short-term memory to the long-term memory, then you retrieve them again the next day, and it makes for better neuronal connections. Yeah. Okay.
So I think that's our last words of wisdom, and we're saying goodbye to you all now.
Okay. And we know that you're going to do well, because our students always do.
They do. They always do well. They always do. So ka kite, see you next time.
Thank you.
Thanks, guys, so much.
Thanks very much. Great course.
Thank you.
Thank you very much.
Thank you so much.
Thank you.
Go the Wallabies next weekend.
Ain't gonna happen, but we can dream.